Hello, everyone. I'm Naveed Shaik. I'm a principal scientist at Gilead Sciences. I will be talking about the physiological-based pharmacokinetic modeling of Lenacapavir, pharmacokinetics, and model validation with drug-drug interactions between Lenacapavir with Voriconazole or Rifampicin. To introduce, Len is a first-in-class capsid inhibitor administered every 6 months. It's approved for treating adults with multi-drug resistant HIV in combination with other antiretrovirals. Len has low solubility, low membrane permeability, and high protein binding. The terminal or the apparent half-life of Len is 10 to 12 days and 8 to 12 weeks following oral and sub-Q administration, respectively. Len is primarily excreted unchanged in feces and is a substrate of PGP, CYP3A, and UGT1A1. DDIs between oral Len and perpetrators of inhibitors as well as inducers of different enzymes and transporters have been previously reported for a phase 1 study. As the magnitude of the DDIs with oral Len administered would be similar to or higher than sub-Q administration, evaluation of DDIs with oral Len represents a conservative approach where with the oral Len, the DDIs will take place at two places, gut as well as at the liver level, whereas with the sub-Q, it will be only at the liver level. The objective of the study was to describe Len PK and predict DDIs with voriconazole and rifampicin using the PBPK model. A mechanistic absorption physiological based PK model of Len was developed using GastroPlus version 9.8.3. The physiochemical and biopharmaceutical properties of Len were mostly from the available in vitro data, and any missing properties were in silico estimates from the model. In vivo data from the healthy volunteer studies were used for the model development and calibration, which consisted of fitting parameters for prediction of Len tissue distribution, clearance, and parameters affecting its dissolution and absorption. Kinetic parameters for Len interactions with cytochrome P450s, UGT, and PGP were fitted against in vivo clinical data from a phase 1 study in healthy volunteers, which evaluated drug-drug interactions of Len with various inhibitors and inducers of enzymes and transporters. PBPK model-based simulations of Len were performed in humans, and all the simulations involved matching the demographics of patients within a cohort of each clinical study utilized in this model. The initial model was calibrated against PK data from selected arms of 3 healthy volunteer studies following either IV or oral administration of Len. The validation was conducted by simulating exposure after 300 milligrams single dose Len administration alone and after pre-treatment with either voriconazole, a known CYP3A inhibitor, and rifampicin, CYP3A, UGT, and PGP inducer, as well as a PGP inhibitor. These simulations were compared with the observed data from a clinical DDI study in healthy volunteers. The developed final model captured the Len profiles reasonably well after IV or oral administration of Len. In the final model, average prediction errors across simulated single IV doses of Len were 26% for Cmax and 13% for AUC infinity. The predicted Cmax was within 0.74 to 1.03 fold of the observed data for IV administration, and predicted AUC infinity was within 1.12 to 1.13 fold of the observed data. For oral administration, average prediction errors across simulated lenacapavir single doses were 22% for the Cmax and 14% for the AUC infinity. The predicted Cmax and the AUC infinity were within 1.09 to 1.34 fold as well as 0.82 to 1.15 fold of the observed data for oral administration. Dose dependency of AUC infinity and lack of food effects on LEN exposure were well captured in this model. The model predicted Cmax and AUC infinity for oral lenacapavir dosing were within 39% of the observed clinical data as shown in the table. There was an overprediction of mean Tmax from 3 to 6 hours to 8 to 9 hours. However, there was no clinically relevant impact of food on lenacapavir PK, and the predicted Cmax and AUC infinity were within 12% of the observed data as shown in the table here. The plots shown here are the model development plots. Top ones are the complete profile in the log scale, and the bottom ones are initial 200 hours in linear scale. The circles are observed data from the study, and solid line is the PBPK model simulated profile for the mean subject in each cohort. For the three dose levels, 50 milligram, 300 milligram, and 900 milligram, the model captured the observed data reasonably well as seen in this plot. Interstudy variability in PK study 300 milligram dose under fasting conditions between the study 4071 as well as the study 4333 was observed, which may be contributing to higher prediction errors for LEN arm shown in this table. The PBPK model was able to predict the impact of Voriconazole and Rifampicin on LEN PK, again highlighted in red. As seen in the table, in the highlighted red for Voriconazole, the top portion, Voriconazole plus lenacapavir interactions, the LEN AUC infinity and Cmax DDI ratios were consistent with the observed clinical data. Similarly, for rifampicin, lenacapavir interactions, the LEN AUC and Cmax DDI ratios were consistent with the observed clinical data in presence of rifampicin. However, the model did not accurately predict the impact of rifampicin pretreatment on LEN Cmax with a prediction error of 59%. The plots shown here are LEN alone, left panel, and LEN in presence of voriconazole in the middle panel, and the right panel is LEN in presence of rifampicin. The squares are observed data from study, and solid line is the PBPK model simulated profile for mean subject in each cohort. As can be seen, there was a reasonable capture of the DDIs. In conclusion, the developed PBPK model captured LEN PK data reasonably well with both IV and oral administration. The final model accurately predicted the impact of voriconazole on LEN exposure, suggesting that the LEN elimination pathway was adequately described. The impact of rifampicin on LEN AUC infinity was well captured. However, the impact on Cmax was not predicted accurately. That might be due to prediction of LEN Cmax with rifampicin, which may require further refinement due to the complexity of a simultaneous contribution of CYP3A, PGP, and UGT1A1. The developed PBPK model can be utilized to predict potential DDIs between LEN and other drugs of clinical relevance. On behalf of the modeling team, we sincerely thank all the participants and the investigators. Medical writing support was provided by Asheville, funded by Gilead Sciences. Thank you.